Okay, good morning. Thank you all for attending online and in person. It's my pleasure to introduce Dr. Soheil Meshinchi, sorry, from Fred Hutchinson Cancer Research Center, who's in Translational Science and Therapeutics. Soheil got his BS from Michigan, University of Michigan, his PhD from there as well, his MD from Wayne State. And then he did PEDS residency at Johns Hopkins, after which he became a fellow at Fred Hutch in pediatric hemonc. He's professor at Department of Pediatrics, Division of Hematology Oncology at UWash, and professor of Translational Science and Therapeutics at Fred Hutch. Soheil's had a number of awards and honors. I'll just mention a few of them: Jose Carreras Leukemia Foundation, Child Health New Investigator Award, Children's Oncology Group Young Investigator Award. And Soheil's been really at the forefront of identifying driving mutations that affect childhood cancer, especially the leukemias. He's been deeply involved at the leadership level in the Children's Oncology Group as vice chair of multiple trials and committees. He's been director of cell and nucleic acid repositories, amongst others, and he's a member of the COG Scientific Council and Senior Science Council for NCI MyeloMatch. His work has been extremely well supported by grants from COG, the NCI, American Cancer Society, various leukemia lymphoma societies, St. Baldrick's, and he's part of a program project grant that John Wagner oversees with Jeff Miller and myself. His research has involved identifying driver mutations and immunotherapy targets, particularly in AML. Some examples of this include recurrent structural alterations in age-specific mutational interactions with NUP98, FLT3-ITD, MLL, T10, ALK, CREBP. And he's published these in the highest level journals: Nature, Nature Genetics, Nature Medicine, Nature Communications, Blood, JCI, JCO, and others. So it's a real pleasure to introduce Soheil to give his talk on the discovery and immunotherapeutic targeting of AML-specific antigens and AML. Thank you for coming, Soheil.
Thank you, Bruce. It's really a pleasure to be back here in University of Minnesota. Seems like I'm here once every 2 or 3 years. A lot of great colleagues and amazing work that's being done. And certainly it's been a privilege to be part of the program project grant that's being done by Bruce, Jeff, and John. So what I'll be talking about is part of our translational work in AML. Again, as a card-carrying pediatric oncologist, our initial focus has been on the pediatric AML area, but as you will see, there's significant extension into not only adult AML, but also other adult malignancies. So just sort of provide a bit of a overview of AML. Overall, the progress in AML and outcomes has been quite disappointing, and this is really highlights really the last several phase 3 trials when we looked at the outcome using the very much a standard of care compared to the experimental therapy. These are phase 3 trials. The top 3 are the adult trials, MRC15, SWOG 106, and most recent SWOG trial, really showing that there's absolutely no difference between standard of care and the experimental arm. So really, this is over almost 3 decades of studies. And the bottom is our pediatric studies that shows the exact same thing, that despite our best intentions, when we really do a randomized phase 3 trial comparing standard of care to an experimental, basically this is a standard of care plus an experimental agent, we've really failed to show any efficacy. And what's more disappointing is the fact our most recent phase 3 trial, it actually showed a difference, but in the wrong direction, where our experimental arm that was, we're hoping to show us a better response, actually provided significantly worse outcome for patients. So this really highlights the fact that we've really reached the limit of what we can do with conventional cytotoxic therapies. And what's really striking is that when you really think about where we are and what we are using as our standard of care, we're using daunorubicin cytarabine that was put in place over 50 years ago. Last November of 2024 was the 50-year anniversary of 7 and 3 or cytarabine daunorubicin. And that is, it remains standard of care in AML, showing that we've really failed to move the field in the last 50 years. Having said that, there have been small successes. We've been able to take some patients with a really, really poor outcome by knowing their genetic and mutational profile. We can actually improve that outcome. For example, the group of patients with the 6/9 translocation, which are highly enriched in a mutation called FLT3-ITD. In early days, at the time that we actually initially identified FLT3-ITDs as a prognostic biomarker, and subsequently we were able to bring in therapeutic TKI to the for therapy. Right now, our outcome really is survival exceeds 90%, something that was less than 15%, not that long ago. So there's an opportunity for us to, if we have a target to go after, we can actually thoughtful approach and really rationally, biologically approach to AML therapy. That's certainly a possibility. One thing that we have been able to do in the interim is to identify patients who are at very high risk of relapse and poor outcome at the time of diagnosis. So what this really shows is that the previous 3 trials, we really did not have very many markers for high-risk disease. But part of our big sequencing project that now we're using for target identification, we were able to actually identify biomarkers that now we've been able to incorporate into our phase 3 trial and try to expand the cohort of patients that are deemed high risk within really a matter of 1 to 2 weeks of diagnosis. So when you look at this, we can actually, at the time of diagnosis, we can identify about a third of the patients that are considered high risk by cytogenetics or molecular alterations. And then we also have about a third that are low risk by this same type of characteristics. And the remaining patients, they're considered standard risk. One thing we have been able to do is actually take these patients that are standard risk and look at their outcome based on initial response by very sensitive methods to detect whether they've actually responded to therapy appropriately by being in deep remission. And that we can actually move these patients, standard risk patients, if they are really negative for any residual disease, to the low risk one, and then those have evidence of disease, they move to high risk. So the way that we use this is to actually take the high-risk patients, actually allocate them to receive stem cell transplant in first CR before they move on, they go on to relapse. And this is just to show you what sort of a gain we have with this. As you can see, patients who are high risk, by transplanting them, the outcome seems to improve. And as you can see in this for disease-free survival, we really have improved. But again, you're talking about still, although the outcome improves somewhat, it is a modest increase at best. So we're still, even by intensifying their therapy and taking to stem cell transplantation in first CR, we are still failing about half of the patients. So really, puts in place that these are really difficult to treat patients, and we really need to have a much more thoughtful approach. Part of the issue becomes, why is it that's been so hard to find, appropriate therapies and therapeutic targets? Because the idea of, because what we're looking for, are there any targets that are uniquely expressed in AML? So what we call tumor-specific antigens. Are there proteins that are expressed only on the leukemic cells and not on normal cells? Because if you are able to identify those, you can eliminate leukemia without really impacting normal hematopoiesis. So right now, the current strategies really focus on CD33, 123, FLT3. These are common antigens. These are expressed in normal hematopoiesis, but also expressed in AML, so what's called cancer-associated or leukemia-associated biomarkers. As you can imagine, if you effectively target these, you're going to cause cytopenias. And if they're really effective, you really need to take them to transplant. If you really eliminate all those cells that express those, and that's exactly what's going on with CD33 and 123 CAR Ts, that you really need to be ready to transplant patients if you have a good response. And then you really can't use them as maintenance therapy because you're going to cause myelosuppression. And frankly, a SWOG trial, when they tried to incorporate Mylotarg, which is the CD33 ADC, they actually had far higher mortality in patients who were treated than observation arm. So, and you also, because of the toxicity profile, you can't really combine. So when you look at I mentioned those three targets. When you compare the expression of these targets, the CD33, 123, and FLT3, 135, what you're seeing is that the expression of CD33, for example, in normal marrow is very similar to what you see in AML. Same with 123 and same with 135. So these are tumor associated, as you can see. They're expressed at a really reasonable amount in a normal cell. So if you target CD33, you're going to kill leukemic cells, but you're also going to get rid of the normal cells. So I'm going to give you an example of mesothelin, which is what we call tumor specific. Compared to these, when you look at the expression of mesothelin in normal marrow, there's absolutely no expression. So this is at the transcript level. So you really have expression, as you can see in AML, which actually overall in some cases is much higher than what you see in CD33, but there's absolutely no expression. That's an example of what we call tumor-specific or AML-restricted targets. So that's basically what we have been looking for, and that's the focus of our studies. So these leukemia-specific targets, or these are basically non-hematopoietic antigens that aberrantly expressed in AML. So technically, these would be ideal targets, expressed in AML, completely silent to normal hematopoiesis. And what we also look for is making sure that this target has very low expression, no expression, other tissues. That would be ideal. Although we can have mechanisms to really pull it into the hematopoietic space by these like logic gating approaches. The other question that becomes that is the cell surface was intracellular target, because strategies are quite different. The cell surface in some ways is easier to access, intracellular more challenging, but there's certainly strategies that we can use. And also these targets, because their expression is only limited to the cancer cells, it also decreases the potential for resistance, such as T-cell exhaustion that we'll talk about a little bit. So when we started this, actually goes back to almost 10 years ago, really the database wasn't there. The only thing was available was the TCGA data that had really a little bit over 100 AML like transcriptome. And we used transcriptome because proteome, it was not really robust, and it's still not a particularly robust mechanism for high throughput screenings. So we started with normal hematopoiesis. We sequenced several hundred normal hematopoietic tissue, normal marrow, CD34 cells, cord blood stem cells, and such. We actually put in place, this is all through initially with a modest NCI funding and then expanded to really privately funded. So we've sequenced over 3,000 patients in all different ages, COG, SWOG, MD Anderson data set. We've also downloaded all available AML databases, TCGA, Beat AML, Lucigene. And we've also downloaded the entirety of the GTEx normal tissue transcriptome. So this is our starting point, 3,000 AML, several hundred normal hematopoietic cells, and all the adult and pediatric downloadable data. So now you're talking about each patient generates about 1 terabyte of data. You have 3,000 for 3,500 patients, normal. So you have a really massive data. I had to learn the new word called petabytes, because terabytes really just is nothing. So then comes the computational part. This is the most critical part of the analysis, that you really need a computational pipeline can actually analyze this data. This is really, we created a collaborative process that included 12 to 15 computational biologists from multiple centers that pulled the data together and analyzed it. So this is something we put in place about 8 years ago. We have weekly meeting every Monday at noon, and we maintain that. And this sort of continues to be a big priority for us, to making sure that the computational pipeline is robust. So the process, just to simplify, to identify our targets, we start with normal hematopoietic tissue. We're looking at the genes that are silent in normal hematopoiesis, or very low expression. And that is our starting point. And take those hematopoietically silent genes, and we look for the expression of those genes in our AML database. And then we filter against the TCGA data as well. So, and then that, process, and this is sort of a graphic representation of starting point. We have, and what we end up is about 200 to 300 potential genes that fit that these are coding genes that are expressed in AML, not in normal hematopoiesis. So the next layer of computational analysis comes, okay, of these genes that we've identified, how many are going to be transmembrane, how many intracellular? That goes through a separate computational process, where each gene that's identified goes through a process trying to identify transmembrane, motif that it has. So a prediction model says this gene, based on the amino acid structure, is predicted to be cell surface or intracellular. And that's a dividing point, because the targeting strategies are quite different. And then once we identify that, then it goes through a verification, making sure that protein, because all of this work to this point has been by transcript. So then genes of interest are verified for the protein expression, and then goes through a preclinical development process. The cell surface proteins can be targeted in multiple different ways: ADCs, BiTEs, NK engagers, and CARs. So we chose eventually CAR-Ts because it's the fastest path to the clinic in an academic center. It's much more challenging to bring ADCs or BiTEs into the clinical space in an academic setting without substantial pharmaceutical help. So the intracellular targets can be targeted in different ways. Our approach has been using TCR mimic antibodies to make CAR-Ts for these, and we'll talk a little bit about that. So basically what we have, I'll show you some data on FOLR1 CAR-T, mesothelin and collect2A. These are our cell surface markers, and for intracellular, we have a PRAME TCR mimic CAR-T, and we have a dual PRAME WT1 that we're working with pharma. So this is our computational output. What we're looking at here, these are the genes these are AML associated, these are AML restricted or AML specific genes that are listed. Red is the expression of that particular transcript in AML, and the light and the dark blue is the normal marrow and peripheral blood stem cells. So as you can see, these have very low to no expression in normal marrow. And and to the right, this is the GTEx data showing expression in normal tissue. So this is for the cell surface, and there's a corresponding one for intracellular targets. So this is our output that we get from our computational pipeline. So I'm gonna switch to one of our initial targets. And actually, when we were starting this process, we're looking for a proof of concept based on an aggressive type of AML with no therapy available, and trying to see if can we identify a target that we can develop and move forward. And we picked this fusion CBF-A2T3 GLIS2. I'm going to call it CBF GLIS for ease. This is a rather newly described entity. It's a cryptic translocation, cryptic fusion, because of the location of on the chromosome that was not amenable to detection by normal karyotype, because they're just literally sitting on the two distal edges of chromosome 16. Technically, it's inversion 16, but it's a bad inversion 16, not the good one that we're used to. So, and this actually came out in really three publications almost simultaneously. Two of them were identified by sequencing and the third one by phenotype. And at the time, we didn't know that they were one and the same. So when we looked at this fusion, this fusion is really uniquely seen in infants and very young children. Basically, we don't have anyone that's diagnosed that's older than age 3 to 4. And it's really, again, 10% of those less than 1 year of age, they have this fusion. And until recently, we didn't know about this particular entity. And what's really interesting is the fact they just don't respond to conventional therapy. It doesn't matter what it is, how intensive it is. They relapse post-transplant very quickly. When we use this particular fusion and put it through our pipeline, this is what we got. We have a number of cell surface proteins, and one of them that we selected was FOLR1. We selected FOLR1 for several reasons. One is that it was actually a known entity in oncology, so there was already some familiarity. Also, we had the sequence we had the antibody sequence from farletuzumab that actually was in clinical trial before. It's a naked antibody for ovarian cancer. So it made it easy for us to make a CAR T using really a published and antibody sequence. As you can see, it looks like there's some other ones that are even better because it's a cleaner background. And we picked it because it was a much faster way to develop a CAR T. So just to provide a couple of more examples. So this is what we're seeing is that we're looking at about over 1,500 patients. And as you can see, the FOLR1 is only expressed in patients with CBF GLIS AML. And when you look to the right a little bit, there's absolutely no expression of the transcript in normal hematopoietic cells, in the CD34 or normal marrow. So it's really a pristine target when it comes to AML. And we were able to verify the expression on the cell surface by flow cytometry. Again, so now we know that we can identify our patients. One thing that we always try to do with our target is the fact that is the expression just irrelevant to the underlying cause of the leukemia, or is there some sort of causal link? So what we did was we took normal cord blood stem cells and transduced it with CBF GLIS fusion transcript. And we actually grew these on EC co-culture that sort of mimics the marrow environment. And what we saw was that literally within a matter of just a few short weeks, we start seeing induction of FOLR1. And by week 12, almost all of the human cord blood cells in that culture were expressing FOLR1. We actually, when we took them and put them in mice, the mice engrafted with AML that was indistinguishable from human leukemia. And what we're seeing in the bottom is that when we looked at the transcript expression of the number of these targets, they are virtually identical in this, the cell culture data and patient data are identical. So by transducing a normal cord blood cell, we basically created a leukemic phenotype that is, so we don't, for this particular fusion, we don't need multiple hits. As you know, AML is known to be, oh, you need multiple hits to get a single fusion is adequate, is sufficient to cause AML. And that's probably why they manifest so early. Because, for example, 821, in most cases, you are born with that clone at the time of birth. Actually, when you check the Guthrie cards, the fusion is there. It takes, at the minimum, 4 years to develop leukemia because you need a second hit. For these babies, it is such a profoundly powerful driver that it takes months after birth for them to demonstrate the disease. So now we have a disease, we have a target that is causally linked to leukemogenesis. So we identified the antibody sequence from the farletuzumab patent literature. We actually cloned it into a backbone. This is a CD19 backbone that was licensed to Juno and then to Celgene for their CD19 CAR. So we swapped out CD19 with FOLR1, and we made a nice CAR T that in our in vitro studies really showed really beautiful efficacy to cause cytolysis in both our cell line model and also our engineered models. So this our initial cell data was compelling, and when we put it into our in a CDX in vivo model, we saw very similarly that they really was highly effective in eliminating leukemia. And then we also generated a PDX model from these are patient samples that engrafted in mice, and we also show high efficacy. So one thing we want to make sure that what we assume that there will be no hematopoietic toxicity is validated. So we actually treated normal marrow and different components of the marrow. First, we looked for the expression of FOLR1 on various normal cells. So the red bar is our engineered cord blood that has high expression of FOLR1. And all of these are different fractions, CD34 positive, hematopoietic stem cell. So all of these, there's absolutely no expression of FOLR1. And then we also looked at the lysis associated. So as we can see, we can actually kill the FOLR1 positive cells, but there's absolutely no cytolysis with the normal cells. In addition, we looked at the ability of our CAR Ts to eliminate, this is a colony forming assay where you incubate cells with your CAR, and if they express the target, then they, then you put them in methylcellulose and you grow individual colonies. So to the right is our colonies after of the engineered CBF Gliss cells with CAR T. As you can see, we've completely eliminated colony formation with our CAR Ts, but we see absolutely no decrease in colony forming in our normal cells. So again, there's absolutely no impact on the normal hematopoiesis with our CAR T. So which is exactly what we were hoping to see. So now we have all the pieces in place, and so we're ready to move into the clinical development. So this is where the problem becomes, getting these assets to the clinic becomes making the vector in a clinical grade, and then developing it, and then having the clinical trial. So where we are right now is that our phase 1 clinical trial. It was cleared by the FDA several months ago, and we actually activated the trial about a month and a half ago, and we enrolled our very first patient. And so we actually infused our T cells, FOLR1 CAR T, first in human into a 3-year-old just yesterday. And this is unfortunately, I couldn't, fortunately, I'm here. Unfortunately, I couldn't be there for that. So this is actually a photograph of when the cells are going in. So one thing that, so this is where limitation of moving assets into clinical trials, especially for rare diseases, becomes limiting. So the funding support for this, although we did have grants to cover part of the early phase, it's really the pace that this really needs to happen. We really moved it from target discovery to FDA clearance in less than 3.5 years. With the grant funding, this would have probably taken twice as much. So the funding support for all the preclinical IND submission was through a project Stella. And again, Stella was our index patient that we identified this fusion in and move it forward. And then you guys probably are familiar with the Children Cancer Research Fund. It's Minnesota, Minneapolis-based organization that they actually stepped up and helped us generate the funds for the actual clinical trial. So, I just wanted to mention those. So this was entirely a private funded process, and I didn't know until recently that this is the very first academic CAR-T at Fred Hutch. So, which was surprising to me, but so this was done entirely without any pharmaceutical support. So then the question, the next thing that we started looking is that, so this is moving forward. We had done all the pieces and when we move into clinical trial, we looked at, this is actually one of my postdocs who pulled this data off. And so what else expresses FOLR1? We knew about some of the adult malignancies, but it was interesting. We actually looked at the pediatric database, and osteosarcoma was one that has almost every osteosarcoma case expresses FOLR1. And then we looked at the pediatric adult, and there's almost every osteosarcoma case expresses it. And this is other malignancies, lung, pancreatic cancer, ovarian cancer, thyroid, and renal cancers. So one thing we actually moved to look at the osteosarcoma as a potential therapeutic target. And so we actually tested our exact same CAR-T that we developed for AML. We incubated osteosarcoma cell line, either with a mock T cells or FOLR1 CAR-T. And as you can see, by 24 hours, almost all of the CAR-treated cells have died. And when you look at the survival, you can see that the mock-treated, they're growing, and then the CAR-treated, they're all dying off. So we moved to an in vivo model. We actually did an intrafemoral injection and developed leukemia. Again, as you can see, the animals who were treated with CAR completely resolved their leukemia. They were alive past day 150. And as you can see in untreated animals or mock-treated animals, it really has a severely destructive process. And osteosarcoma can also present as a metastatic disease to the lungs. We also generated a metastatic model, and we saw exactly the same thing. So we really do have. So based on this data, now we have a second trial in osteosarcoma that we're in the process of. We made a second separate batch of vector at IU, and it's moving through the preclinical evaluation and engineering runs. With the trial activation, we're hoping that's going to be end of the year, maybe early next year. And given high prevalence of this target in other solid tumors, we're actually trying to see whether we can actually open it up to truly a basket trial where we can actually enroll other malignancies and trying to assess at least safety and efficacy. The solid tumor world has been quite challenging when it comes to really getting CAR-Ts to work. So we'll see how that moves. But the osteosarcoma, we're quite excited. And by the way, so that is a similar process moving forward. I'm doing a sabbatical enrollment for Franco Lucatelli's group. So he's actually moving FOLR1 into all malignancies as a true basket trial. They're able to do that much faster. So that's also moving in. So that's going to be available across Europe as they're able to deliver it to multiple centers. So we developed this CAR for this tiny fraction. We're talking about 20 patients a year, maybe. And so we're moving into 1,000 patients in osteosarcoma, but you can really see the potential that if it's really effective, and the field is really moving away from treating a single disease to really going after targets, and actually we broaden targets from solid tumor and AML. So there's at least a possibility that extend from AML. So that's our sort of proof of principle, and we've made it to our first patient. So the question is there another subset of patients that we can actually have a similar approach? So the other area of high need and unmet need is KMT2A rearranged AML. This is leukemia that is highly prevalent in children with newly diagnosed AML, but it also highly prevalent and highly refractory disease in what's referred to as secondary AML in adults, where they're treated for one malignancy, for breast cancer or colon cancer, and then a couple, 3 years later, they develop AML, and that's usually driven by KMT2A rearrangement. So, the problem is that even with most intensive therapy, the outcome is quite poor. So this is when we look at the outcome for with de novo AML, so newly diagnosed AML with KMT2A rearrange, you're talking about the survival really drops down to 25–30%. It's much worse in secondary AML. And this is when you look at the adults with secondary AML post exposure to other cytotoxic therapies, it's really an incurable disease. So it's really, really challenging. And right now, the field is really inundated by companies who are actually developing this, what's called a menin inhibitor. Menin is one of the genes in this complex, the KMT2A complex. So 6 companies are really developing menin inhibitors. Syndax actually got approval for their drug with a 23% CR rate that is transient. Actually, almost every patient gets into CR. If they're not transplanted, they relapse within 3 to 4 months. So as you can see, there's efficacy, but very short-lived. Almost everyone who responds, if they're not really moved to transplant, they relapse. And even after transplant, many of them relapse. So we feel that this is an area of great need, both on the pediatric, but also adult side. So now we basically took our computational database and asked the exact same question. Are there targets that are expressed in KMT2A that are not expressed in AML? And so as you can see, we have multiple targets, and the top one is CLEC2A. I'm going to talk about CLEC2A. Mesothelin is another one that I mentioned earlier. And but CLEC2A is really, as you look at it, it is the expression is actually almost non-existent in normal marrow, expressed in AML. But look at this. This is really a clean expression in other tissue. So, and even within KMT2A, KMT2A has lots of different fusion partners. So when you look at this, you can see enriched in MLL-T10 and MLL-T. These are actually some of the highest risk KMT2A fusion partners. So when you look at this, you can see that this is the population that we're looking for. And KMT2A is certainly in that group of basically no expression in normal marrow, but it has high expression in AML. So again, so this is across the board for pediatric, and this is adult data. There's no expression of the transcript in AML in normal marrow CD34 positive cells. And this is the KMT2A rearranged population. That's majority of them, they express it. And again, this is simply looking at the high-risk group. This is MLL-T10, which is very high risk, and this is MLL-T4 high risk, but overwhelming majority of these patients express the target. What's interesting is this population here, that they don't have the fusion, but they express the FOLR1. And this is a new observation that we made, that's monosomy 7. And those who treat and transplant AML, they know that that is a particularly challenging group of patients, that no matter what you do, our outcome for monosomy 7 hasn't really changed in 40 years. So this is actually surprisingly a surprising group of patients that now there's a possibility for targeting. So now let's look at the expression in normal tissue. So this is the data from GTEx. So looking at the expression in various tissue, you can see there's absolutely no expression in any other tissue. And the only thing that's some expression is CLEC2A, is the skin. And this really brings attracts everyone's attention because, oh my gosh, you're gonna, slough off the skin. So, but when you compare that to expression in the skin of various targets that are either in clinical trial or actually FDA approved, it's actually very modest to very low expression. And and we've actually done studies that, we've actually have skin cells, in culture that we've treated that shows absolutely no toxicity. So, and the other question that, again, going back to the question of is there causal link? So we're, while we're trying to induce our expression of CLEC2A in our cord blood system, we actually collaborated with Steve Henikoff's lab that they actually created the next generation of ChIP-seq they call cut and tag or cut and run, where basically they're looking at binding of fusion oncoproteins to various promoters. So this is sort of a next generation of ChIP-seq. So what we were able to show is that direct binding of KMT2A fusion oncoprotein directly to the CLEC2A promoter. So this was really showing, again, direct and causal link between the fusion and induction of the antibody of interest, and this basically binding score that they generate, and the higher the binding score, the higher the CLEC2A expression. So this is, again, so we want to make sure that we are comfortable that there's some link between the fusion and the induction of that, and that's what we have. And we're actually generating normal cord blood transduced with a fusion to show similar expression as we did for FOLR1. So, here we show FOLR1 expression in KMT2A rearranged cell lines and lack of expression in cord blood. Again, in primary patient samples, we show expression in KMT2A and monosomy 7. And we've actually generated the PDX models for our study. So this is a monosomy 7 PDX model that expresses CLEC2A, and we actually have multiple KMT2A rearranged PDX model for our experiment. So from that standpoint, we've discussed this. If there's any interest in PDX models, we've just generated it for our studies, and these are easily available with a simple MTA. And we've actually transferred a number of these to a PDX repository in Texas Children's. So the only problem was the fact that actually if you do a PubMed search for CLEC2A, you'll get a total of 10 articles, and there's not a lot known. There's not a lot of information. There's no antibody sequence that we can use. So we had to develop our own antibody. So this is in collaboration with Wei Li's group in University of Pittsburgh. They actually made fully human antibodies for us. They gave us all together, they probably generated over 100, and they gave us 24, and we tested them, and we had actually one that really seemed to, two that actually worked really well, but so we used one for our CAR T. So we actually cloned the 3H10 binder to our to the exact same vector that we had, and we saw beautiful cytotoxicity, as you can see, in our ex vivo model. And again, we tested the cytokine release, and we also showed no efficacy in normal cells. So this simply shows our CDX model for mice that were treated with our CLEC2A CAR-T. As you can see, the treated animals, regardless of whether it's a high or low disease burden, they seem to have quite nice efficacy and to eliminate the disease. And then we also did the actually, as you can see, that this is just simply the IVUS imaging results and what is in the bone marrow of treated and untreated animals. So from so really high efficacy that we're seeing with our CAR-T. And very similar data in our PDX model. As you can see, our PDX model completely eliminated the CAR-T, completely eliminated leukemia in our PDX model, in the same model that by day 36, all animals were dead from the disease. So again, we're at a similar place that we are ready for advancing this into clinical trial setting. And we deal with the exact same issue that the algorithm is that if you want something to move into clinical trial, you have to go find a pharma partner, and that's been challenging. So, it took us a while to move it forward. However, that's one of the reasons I'm doing my sabbatical in Rome with Franco Lucatelli's group. Within literally 2 weeks of arriving in Rome, this was prioritized through their pipeline. So this actually, they already have the CLEC2A GMP vector in hand going through. It's going to be going through the engineering runs in the next month or two with a plan to activate a trial in Rome. Actually, it's going to be delivered EU by the end of the year. So that's a timeline. Meanwhile, we have been able to get some commitment about moving it forward in Seattle, but that's going to be delayed. So this actually, our second CAR-T that's actually moving into clinical space, hopefully in the next, probably in the upcoming year in Europe and next year in US. But you appreciate that bringing it through the preclinical space can be quite quick. It's taking the next step is limited by resources, but we've sort of been able to navigate that. So I'm going to spend a couple of minutes the strategies to go after intracellular targets using CAR-Ts, because that's the issue. Because intracellular targets, they're not quite accessible by antibody binding and CAR-Ts. So PRAME is one that we use because the antibody for the PRAME peptide that is presented on the cell surface through the HLA molecules was available. So a company called Eureka, they had developed an antibody that recognized, so basically all the intracellular proteins are processed and then they presented on the cell surface as in a part of the MHC complex. So what the company had done was actually quite brilliant. They actually tried to see if they can actually generate antibody that recognizes the peptide, the broken down piece of protein, in the context of that HLA groove. And they had an antibody that recognized, so this is HLA restricted, so only half the patients with HLA-A2 would be able to benefit from this. So we took that antibody and we created a CAR-T for PRAME in the HLA context. And so the PRAME is another beautiful target. It is expressed in AML, adult and pediatrics, and various fusion groups. But when you look at the expression in a normal tissue, it is absolutely no expression unless it's in the testes. This actually goes into what's called a cancer testis antigen. It's a sanctuary site that there are very, very few proteins that are expressed only in the testes and nowhere else. So it's a sort of an immune sanctuary site. So it's actually a classic cancer antigen that people have been trying to target. we did the exact same thing we did with all the other ones. We created a PRAME CAR T that showed beautiful cytotoxicity in our culture model, and also incredible efficacy in our both the CDX and the PDX model. So it's really quite remarkable. And so this is actually, we're in the process of trying to move this into clinical space. So right now we're trying to decide between the standard PRAME CAR T versus what's the company that we're working with. They have an Artemis platform where they can actually put binders, for antibody and a secondary co-stim domain that can provide either enhancer or AND gating. So we have generated a PRAME CD33 AND gated, where let me actually quickly. So basically, what you're seeing is that in CD33 positive cell lines, you're seeing beautiful efficacy. If a CD33 negative or knockout, you lose all activity. So this is any tissue. This really increases tissue specificity. So, for example, so it eliminates any sort of a non-specific binding and non-toxicity. So that is actually where we are in the process of deciding whether to use the Artemis platform or the standard PRAME CAR T. So that is we're hoping that that's actually be in the clinical space quickly, because of the fact that if we use Artemis platform, that's going to be a much faster path. So overall, our FOLR1 is in the clinic. Mesothelin and CLEC2A, they're really mesothelin. We have a trial that we're submitting an IND by the end of the year. CLEC2A, it's going to be active in Europe by the end of the year, and in US probably early part of middle of next year. We have PRAME that's likely moving into clinical development, and these are some of our newer targets. Actually, 276 B7H3. You guys have one that's it's a terrific target. CSPG4 is one of our really it's a great target to have. So I think all of these are moving forward. So I'm hoping that I've been able to sort of convince that there is actually AML-specific targets that we can go after. It's not quite as broad as one would like. We'd like to have one CAR that cures all AML. So far, in the last 50 years, we failed to do that. I don't think that's going to happen. We can the transition to solid malignancies pretty effectively with some of these targets. And then this sort of a dual targeting and as well as OR gating can really help us broaden both specificity and sensitivity. Anyway, and that's thank you for all. That's the lab, and that's the lab folks in the corner. So, all right, thank you.
Thanks, Soheil, for a very comprehensive talk. I wanted to ask you a few questions. For the folate receptor, do you think that there's any role of folate in the pregnancy that helps support I'm sure you've had that conversation, but since it's happened so early, it must have a proliferative advantage that might be driven by folate.
Are you talking about whether folate is helping propagate the leukemia? Well, that's the question that we're asked by almost every parent. That it's kind of hard for me to my guess is not really. Having said that, it's not just the simple expression of the folic acid receptor. It seems like the folate pathway is activated. In fact, we've actually treated a few patients with methotrexate because we had nothing else. Our very first patient was a magical response, and the next 4 had no response. So the question is like, is the folate pathway is activated, and that's one thing that we're actually looking at that.
So where do you think the second hit hypothesis stands with all of these different mutations? Is some of them only need one because they're potent
Absolutely. I
drivers?
think it's just driver because, this the two-hit hypothesis, I mean, Gary Gelland sort of brought the class 1, class 2. I mean, using A21 translocation was on A21, absolutely. When you look at the AML across the board for A21, A21 has the highest number of mutations of anything else. And also it takes 4 years minimum for patients who have that fusion to develop AML. For this, I mean, we put it in cord blood stem cell, and within weeks they have AML phenotype. So I think that it's variable. I think that the two-hit hypothesis, I don't think it's generalizable to all AML. In this case, it's one hit is sufficient. It's such a powerful driver. In A21, absolutely. If you put it in A21, same with actually NOPN-ST1. We put NOPN-ST1 into cord blood stem cell. We had no phenotype. You put ITD, then you have an expansion.
My last question is, could you comment on using CD45 as a pan hematological malignancy targeting approach from
So
Soheil?
CD45, I think that if you're targeting for, like, for example, for conditioning, so if you want to basically eliminate stem cells, I think 45 would work. We're actually planning on using 45 as a AND gating to pull in, because CD33 is that, because of all the polymorphisms and splice junctions, not the ideal target for pulling into the hematopoietic space. So FOLR1, I mean, CD45 can theoretically be a great sort of a pan leukemia target. I think, but you need to be ready to transplant patient or use it as a part of the conditioning regimen.
Unless you do what Soheil and Lucas Jeter did, which is knock out CD45 on the.
Well, then that's the question. If you don't need CD45 for hematopoiesis, so that's basically what happened with CD33. So you have to engineer it to knock it out. I think that's certainly a possibility.
Okay, we've got a question from Dr. E online. Can you comment on the activity of the FAL-R1 ADC? I'll try to say this, mirvetuximab in leukemia. What would be advantages of CAR T over ADC in solid tumors?
Yeah. So there's mirvetuximab, there's luveltamab, and there's another one. So we actually looked at all of those as part of that was our short-term plan for helping these kids. So we actually tested mirvetuximab, Sutro's drug, and Elucida's drug. Sutro and Elucida had great efficacy. In the same preclinical testing, we saw no efficacy to mirvetuximab. Having said, and then Sutro, the STRO-002, has had remarkable efficacy, and we've treated patients with CBF GLIS, but we've treated about 50 of them on a compassionate use basis. We actually moved the drug into clinical trial. Unfortunately, company, as of literally 2 weeks ago, deprioritized the drug. And with the lack of other options for patients, I think mirvetuximab is worth trying, although we saw no efficacy in our preclinical models. You could probably see something. Genmab has one that has really good payload. That's possible. Elucida, unfortunately, is no longer in existence. So CAR T it's a one and done. You have one infusion. If it works, you're done. With these drugs, we've actually had patients on STRO-002 that have been on it for 2 years, and they've responded, but it's a 2-year. And those who relapse, they're FOLR1 negative. So advantage of CAR, again, it's a one and done. So if it works, then you're done. And if it doesn't work, you will know within 3 to 4 weeks. So.
This is really fascinating. And I took a quick little detour when you mentioned FOLR1 in osteosarcoma, because we work on dog osteosarcoma. And I wanted to check and see if the antigen was expressed. And it turns out that it's not. So I wonder, given that most of the samples of osteo that you probably have are pediatric or maybe young adult, and the tumors that the dogs get are age associated, so they get this when they're really old. And what you see in terms of the selection in the FOLR1 leukemias, do you think that it has something to do with having expression of that particular oncogene in an early developmental stage rather than purely a histological tissue type? So maybe the developmental stage more important than the actual tumor histology?
Well, that's a really good question. I think developmental stage makes a huge difference in leukemogenesis. For, in all honesty, a lot of work that's been done in trying to see if a fusion or a genetic event leads to a phenotype. When you put it in adult hematopoietic stem cell, you don't get a phenotype. You put it in cord blood or liver hematopoietic cells, you get an incredible phenotype. So I think development stage is absolutely, a determinant of disease evolution. There's, whatever it is, and that's actually been shown by multiple, groups that actually the early cells, from young, cord blood or liver, they transform differently than adult hematopoietic stem cells. Regarding osteosarcoma, that I know that folic acid pathway is substantially involved in osteosarcoma pathophysiology. Exactly how it's different. So when we looked at adult osteosarcomas at the Hutch, we do see FOLR1 expression. Having said that, I don't know what the variation histology and FOLR1 expression is. When we look at the FOLR1 across the board, majority of the patient expressed it, but they all had similar histology. So right now we're collecting specimens at the Hutch from trying to determine if like adult osteosarcoma, different different phenotypes and subtypes of osteosarcoma express FOLR1 or not. That's something that we don't know. We're hoping to figure that out because that's going to be the eligibility determinant.
All right, I think we are out of time today, so let's say thank you one more time.
Okay, thank you.